Hi everyone, I'm going to be discussing the four macromolecules of life, and I'm going to close my little window here so that it doesn't block the slides because I find that really annoying when I'm watching presentations. Okay, so let's start. The four macromolecules are going to be carbohydrates, proteins, nucleic acids, and lipids. Now, I want to talk a little bit about modeling. So, when you see these macromolecules depicted, they're actually going to oftentimes be drawn shorthand, like this one that you see here of a fatty acid, and it just looks like a squiggly, jaggedy line. But in actuality, what it's not showing is that at every corner, at every point, and at every end, there is a carbon. And what that looks like is like this. And then off of the carbons are actually hydrogens as well. So those are also not depicted here. So what I want you to know is that the squiggly line itself, so let's go back to this first one. There's a carbon right here, and the line in between this angle and this end, or this angle and this end, the line in between represents a covalent bond. And now remember that carbon can make up to 4 covalent bonds. And if you only see one here off of this carbon and another one here, that leaves 2 more bonds that need to be fulfilled. And so there's hydrogens here. And you can see that depicted here. So this one carbon is bonded to hydrogen, so that's 2 bonds. And then the third bond is to another carbon, and the fourth one is to another carbon down here. And so whenever you see these shorthanded models of molecules, just assume that there's carbons and hydrogens there that you don't see. And finally, here's a space-filling model depicting the exact same thing. So the first one is a fatty acid, and then the same fatty acid is shown in the middle. And then finally, there's a space-filling model of the exact same thing, which is this fatty acid. And in that case, you're supposed to know that the gray depicts the carbon, and the white circles depict the hydrogens, and the red the oxygens. So just so you know. So let's go back to this image right here of these molecules. So these models of these molecules are also written in shorthand, and what you don't see again are the carbons. I'm just going to talk about this first one right here, which is carbohydrates, and this is a disaccharide here. So that means there's one saccharide, there's one monomer attached to another monomer of carbohydrate, and all the carbons are missing. They're not depicted, but they are there because this is in shorthand. So what does that look like? Well, if we go to this monomer right here, we're going to start at this corner, and this is the carbons are actually numbered, and they're referenced by their position in the molecule closest to this oxygen right here. So let's start with, here's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and remember carbon is CHO, C6H12O6. So where's that? If it's C6, where's the other carbon? And that carbon's right here. So 1, 2, 3, 4, 5, 6. And if we look at the second monomer of a saccharide here, it's going to be the same deal. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and at the top is the other carbon. So I'm going to briefly just show you the same things happening here with the proteins, is that you don't see where the carbons are, but they are there. So here, this carbon right here that I just showed you, see how the lines, remember, represent covalent bonds. And so this carbon has a double covalent bond here. So that's 2, 3, and 4. So this carbon doesn't actually have any hydrogens coming off of it. And so that even if it did, it may or may not depict it. It just depends on how many bonds carbon has. But carbon is always bonded with 4 covalent bonds. And then for the next one, same thing. So here, this carbon has 3 bonds to it, which means there's a hydrogen off of it that's not being shown. And the same is going to be true for the nucleic acids. And then finally, the lipids down below. They're just not showing all the carbons, but the carbons do exist. All right, so let's review macromolecules very briefly. Small organic molecules are called monomers, and when they join together, they form large molecules called polymers. So macromolecule, which is the polymer here, there are 4 major classes of macromolecules, and these are the biological molecules that make up all of life that as we know it on Earth: carbohydrates, lipids, proteins, and nucleic acids. So, what are polymers? Polymers are bigger chains of monomers that are attached together through a process called polymerization. So, an example I want to give you is maybe you're building a Lego tower. Each one of the single Legos would be considered monomers. And then the giant tower that you made, when you attached all those monomers of Legos, individual Legos together, that giant tower is going to be the polymer. And your hands are actually the enzymes. Enzymes are facilitating this whole process of converting monomers to polymers and vice versa. So, how do you build a polymer? Well, you need an enzyme and you need energy. So, to build a polymer, it's called synthesis. And what you're doing is you're joining monomers by taking water out. So, you're taking the hydrogen ion from one side of one monomer and the hydroxide from another. And the enzyme is facilitating this process. Um, and it's synthesis by removing water. So we call that dehydration synthesis. Because if you were to dehydrate something, you're taking the water out of it. And in this case, dehydration synthesis is taking water out to build a polymer. Now, one monomer donates the hydrogen hydroxide ion, and then the other is donating the hydrogen ion. And together, the water is removed. So this requires energy, like I said again, and enzymes are facilitating this action. Another term for dehydration synthesis is going to be a condensation reaction. And so if you're familiar with condensation, let's say like you have a really cold drink out and then you leave it out for a little while and then water beats up on the surface. So where did that water come from? It's either from the environment or from the drink itself, but generally it's so cold or the difference in temperature is causing water to bead on the surface, so condensing out. So condensation reaction is the same term we use when we talk about dehydration synthesis. Okay, so how do you break down a polymer? Well, another term for breaking down a polymer is digestion. So when do you need to break down a polymer? Well, every time you eat, when you're eating food, you're eating the macromolecules. You're eating proteins, which are polypeptide chains. You're eating polysaccharides, which are long chains of carbohydrates. You're also eating nucleic acids, so either DNA and RNA, because you eat things that used to be alive, and all living things had genetic material in it. And then you're also eating lipids, so fats. And fats are long chains of hydrocarbons. So we need to break them down for a couple of reasons. One is we need the actual building material to make more of us. So for growth and development, and also just for repair and maintenance. So like let's say I'm not actually growing any taller, my bones aren't growing any taller, but my cells wear out and then they die, and then I need to replace them with new parts. So I need to eat for that. And then the other thing I need to eat for is energy. So the energy that's held in the bonds of the chemical bonds of those molecules, I need to digest them so I can get energy to keep my body alive. So how does that work? So to break down a polymer and turn it into the monomers, you have to use water. So you're actually putting in water. And when you put in water, it's the reverse of dehydration synthesis. So it's actually like the reverse reaction. And that it cleaves off one monomer at a time. And it also, when the water is split, it's split into a hydrogen ion and a hydroxide ion. And that's attached to either side of the monomers. And it also requires enzymes. And actually breaking polymers down into monomers releases energy. The other thing I want you to know is that this reaction is called hydrolysis. So hydro means water, and lysing is to break something apart. So hydrolysis is literally breaking apart with water. And that's another term that we use for it is digestion. So let's talk a little bit about polymers. Polymers are long molecules that are built by linking repeating building blocks in a chain, kind of like the example that I said about building a long tower with Legos. Or if you think of like a chain with actual links to it, like either a necklace or a chain that you close a fence with. And it's created by dehydration synthesis, and it's broken apart by hydrolysis. So those two terms you do need to know. And what does that mean? One is taking out water and the other one is putting in water. And both those reactions require enzymes in order to occur rapidly. So these reactions would eventually happen on their own, but the rate at which we need them to happen would be too slow without enzymes, and we would die because we, you know, like millions of chemical reactions are happening in our body every second. And the speed at which they need to happen in order to keep us alive requires enzymes to do that. So let's talk about functional groups. So functional groups are actually super important, and it's important for you to recognize them so that you know their behavior. Um, so basically, biological molecules are just really long skeletal chains of carbon and hydrogen. And then they are modified with functional groups. And what these functional groups do is, well, first of all, these functional groups um, they always behave the same. So they give organic molecules very distinctive properties, and those properties are always the same in terms of like solubility, reactivity, etc. And here are the 6 that you should know: hydroxyl, carbonyl, carboxyl, amino, sulfohydryl, the phosphate group, and they affect reactivity. So how the reaction is going to occur. And remember, reactions are occurring millions upon millions a second. So they also make hydrocarbons hydrophilic, and that's actually super important because remember that we are mostly made of water, and so we need these molecules in our body to be water soluble in order to interact properly in our bodies. So this is a perfect classic example of what functional groups do and how they make such a difference. So if you look at these 2 images at the bottom, we have a female lion and we have a male lion. And what makes female lions and male lions has to do a lot with steroids. And so steroids are actually a class of, they come from lipids. So the backbone or the skeletal structure comes from lipids, and the basic structure of female and male hormones are actually identical. And this is a little bit of a side, but oftentimes if you are severely malnourished or underweight, if you've lost a lot of your fat, it actually can affect your hormonal balance. And I had a colleague, and she was having a really hard time with ovulation. She had really irregular ovulation, and she was vegan. And she met with her doctor, and her doctor was saying, well, you actually need to increase some fat in your diet. And that doesn't necessarily mean that, you have to break away from your vegan diet, but it would be easier to get some of the different macromolecules in your body if you were to change it up a little bit. And that actually helped her with regular ovulation. But and part of that has to do with how the hormones are in your body interacting. And remember, hormones are lipid based. So if we look at these hormones, estradiol and testosterone, estradiol is found in a female lion and testosterone is found in a male lion. And those steroids or hormones in this case cause them to behave very differently. If you notice that the molecules have the same identical backbone, their skeletal backbone is the same, or carbon backbone. However, the functional groups that are attached to them make them behave very differently in how they interact in the body. So the estradiol has a hydroxyl on it, and then the testosterone has a methyl group, and then the carboxyl group on it. Okay, so, and of course, like I said, the functional groups result in different effects on your body. Next up, we're going to talk about the different kinds of macromolecule groups.